Good afternoon and good evening. I'm Deb Coleman, Chair of the Board of the Damon Runyon Cancer Research Foundation. Welcome to our virtual Toast to Science. We are honored and immensely thankful to have two very special guests, Dr. Harold Varmus and Dr. Bill Kaelin, who will discuss with three of our wonderful Damon Runyon scientists and with us how best to advance cancer research. Both Dr. Varmus and Dr. Kaelin have dedicated their careers to cancer research, and both are recipients of the Nobel Prize in Medicine or Physiology. Who better to lead this important discussion? Damon Runyon has a particular approach to cancer research. Our methodology is simple: identify the best and most innovative young thinkers and support their bold cancer research goals in the clinics, in the labs, and at computers. Damon Runyon was founded by Walter Winchell, a character, a widely syndicated columnist and radio host, upon the death of his friend, Damon Runyon of Guys and Dolls fame. Since then, Damon Runyon alums have gone on to receive some of the most prestigious awards in science, including 12 Nobel Prizes and 15 Lasker Awards. These prizes, though, are not ends in themselves, but really are indicative of the types of breakthrough science that can be achieved by supporting the work of the brightest and most innovative young minds. Unlike the complex and dynamic mechanisms of cancers, Damon Runyon's mechanic is in fact quite simple. There are three parts. First, our backbone. This is comprised of our amazing donors and other supporters, including all of you, our dedicated staff, especially Kim Kubert and her team who coordinated this program, the Broadway community, which has, dating back to Walter Winchell's days, been a stalwart supporter of our efforts, and the non-scientist members of our board, who not only are extremely generous, but also lend their considerable professional expertise to crafting and supporting the most impactful award programs. Thank you to all of you. A special thank you to Alan Leventhal, who led our board over the last 10 years with wisdom and an unwavering commitment to excellence. Alan set the standard for stewardship, and we are enduringly grateful. The second part of our mechanic, our brain, I often refer to this as our secret sauce. These are the renowned cancer scientists and physicians who sit on our board and comprise our award selection committees. Currently, a third of the board is comprised of scientists. Over 60 additional scientists volunteer their time to review the work of, interview, select, and mentor awardees. These scientists are leaders in cancer research and therapy development across the US and drive the agenda of Damon Runyon. Thank you very much to all of you. The third part of our mechanic, our heart. The 212 audacious young scientists currently being supported by Damon Runyon, and the many Damon Runyon alums, all of whom are contributing in their own way to our central objective, improving treatments for and curing cancer. We are rooting for you all. So speaking of Damon Runyon scientists, it is now my great pleasure to introduce the president and CEO of Damon Runyon, Young Lee. Young was herself a Damon Runyon Fellow. After working in cancer research, she served as Damon Runyon's chief scientific officer under the indomitable Lorraine Egan. It should be noted that during the period from 2009 through 2020, a period with Alan Leventhal, Lorraine, and Young at the helm, Damon Runyon increased its annual award programs from 9 to 20 million, and the number of scientists supported each year from 118 to 212. As a cancer researcher, Young is uniquely qualified to lead Damon Runyon forward. We are grateful and excited for her leadership, and I'm really excited for you to hear from her now.
Thank you so much, Deb, for that very kind introduction. We are so glad that you're joining us from wherever you may be. While we're not able to be together in person, we're thrilled to have you participating today so that we can celebrate our amazing Damon Runyon scientists. This year has been challenging on so many levels, and we have all had to reimagine many parts of our lives and work due to COVID-19. Despite the many challenges, our scientists remained dedicated to their extremely important work. They had to be especially resilient, during the lab shutdowns this spring and summer, trying to keep their research going while also focusing on work that could be done remotely, including analyzing data or writing up their work for publication. Some were even able to pivot their research efforts in ways that specifically contributed to knowledge about the SARS-CoV-2 virus and to new treatment options for COVID-19 disease. As an organization, Damon Runyon has likewise continued our work to enable critical support of the next generation of leaders in cancer research. Our mission is funding brave and bold. We have not hesitated and continue to invest generously in the most brilliant young minds. The amazing young scientists we fund focus on all areas of research across all cancers. Our portfolio of award programs supports the full range from fundamental basic discovery research through our fellowship and Dale Fry Award for breakthrough scientists to ambitious, high risk, high reward projects through the Innovation Award to more translational research that bridges the gap from lab to patient through our clinical investigator and physician scientist training awards. Our newest award is the Quantitative Biology Fellowship, which supports the training of computational quantitative scientists in lab-based cancer biology. Our scientists are working in leading research centers across the nation to find cures for all types of cancer. Since our founding in 1946, they have been at the forefront of key advances, from first establishing the link between smoking and lung cancer, and the first cure of a solid tumor with chemotherapy, to more recent advances, including the development of CRISPR gene editing technology, novel immunotherapies that harness the body's own immune system to attack cancers, and targeted therapies that can treat cancers more effectively with fewer side effects. It's now my absolute pleasure to introduce our panel. We are honored to have today's conversation led by two extraordinary scientists, Drs. Bill Kaelin and Harold Varmus. Both are physician scientists, specially trained in the clinic with a focus on patient care. Each developed a deep interest in laboratory research to better understand the biological mechanisms underlying cancer and other diseases. Both have been recognized for their spectacular scientific accomplishments, with prizes and accolades too numerous to list now, including the Lasker Award and notably the Nobel Prize in Physiology or Medicine. Dr. Kaelin is a professor of medicine at Harvard University and the Dana-Farber Cancer Institute, and his laboratory studies tumor suppressor proteins. He received the Nobel Prize for unraveling a molecular mechanism that explains how cells sense and adapt to varying levels of oxygen. These findings could lead to new therapeutics for a wide range of disorders, including cancer, cardiovascular disease, anemia, and macular degeneration. Dr. Varmus is a professor of medicine at the Meyer Cancer Center of Weill Cornell Medicine. He was previously director of the National Cancer Institute, president of Memorial Sloan Kettering Cancer Center, director of the National Institutes of Health, and started his academic career at the University of California at San Francisco. His laboratory studies the molecular mechanisms of oncogenesis, or how normal healthy cells are transformed into cancer cells. He received the Nobel Prize for his seminal studies on the genetic basis of cancer. Not only are their individual accomplishments groundbreaking and paradigm shifting, but both Bill and Harold have demonstrated a unique commitment to mentoring and fostering the next generation of physician scientists and cancer researchers. Notably, Dr. Varmus has mentored 10 Damon Runyon scientists, 2 of which are currently in his lab. Dr. Kaelin has a deep involvement with Damon Runyon as the current vice chair of our scientific programs and has led the selection committee and development of 2 award programs focused on supporting physician scientists. He is currently mentoring 2 Damon Runyon scientists in his lab. Bill and Harold are joined today by 3 current Damon Runyon scientists. Lindsay Case, PhD, who is a recipient of the Dale Fry Award for Breakthrough Scientists. She was a Damon Runyon Fellow at the University of Texas Southwestern in Dallas, and she will be starting her own lab at MIT in the new year. Jay Choi, MD, PhD, is a Damon Runyon clinical investigator at Northwestern University outside of Chicago. And Kavita Saran, MD, PhD, is also a Damon Runyon clinical investigator, and she's based at Stanford University. Thank you so much to our panelists for joining us for today's program, and I'm pleased to now turn it over to Bill, Harold, and our three Damon Runyon scientists.
Wonderful. Thank you very much, Young, for that very nice introduction. It's a pleasure to be with you all here virtually. You know, maybe I'll start with Harold. I mean, given that you were the director of the National Institutes of Health and the National Cancer Institute and were the president of the Memorial Sloan Kettering, and as we just heard, you've had, I think, 10 Damon Runyon awardees over the course of time. You're probably the best person on the planet to articulate why is the Damon Runyon important? What role does it play?
Well, Bill, thanks very much for the question, and Young, thanks very much for having me here today. All postdoctoral fellowships that support talented trainees are important, but one of the sweet spots in the Damon Runyon fellowships is that there is a tendency to think about the relationship between a trainee and a mentor as a kind of marriage. It isn't just a temporary relationship, it's one that lasts throughout one's career. I say this not just as someone who has been the mentor for a number of Damon Runyon Fellows, but as also someone who had 3 terrific experiences as a mentee, not, I admit, under Damon Runyon's aegis, but just at the NIH, where I first learned to do research as a young physician who really hadn't done any research before and was at sea. Partly it was as someone who recently went to UCSF about 50 years ago to start working with Mike Bishop. And then as a young faculty member, just learning a lot about tumor virology from Peter Vogt. These 3 people, still wonderful, have been close colleagues of mine for 50 years as a result of the healthy atmosphere that arose through the mentee-mentor relationship. And that when a Damon Runyon Fellowship works well, it works well because there is a harmony between the mentor and the mentee. And what's transmitted is not just a willingness to allow the fellow to use reagents and to get a good letter of reference for the next position, but also what's conveyed is an attitude towards science, how important model systems are, how important it is to recognize when a new technology changes the range of questions that you can address, how important it is to understand that sometimes the focus should not be on application of the science we're doing, but trying to understand fundamental mechanisms the way your research and mine have proceeded. In addition, an ideal relationship that can be created by a good pairing of mentor and mentee is one that conveys not just the beauty of science, but also its quality as a public good. That science is for everybody. We're paid, whether it's by Damon Runyon or the US government, the NIH, other donors, to do things that enhance the human condition. And communicating that, the importance of sharing, of collaborating, of working in a harmonious relationship with other scientists around the world are features that I think are encouraged by enlightened fellowship programs of the kind that Damon Runyon illustrate.
Thank you. Well, maybe on that note, since we do have 3 of our wonderful awardees with us, I'm going to ask each of them to give us just a brief snapshot of what they're working on and what they're thinking about. So I think we're going to start with Lindsay Case, who, as we heard, is a fellow at UT Southwestern. And I think Young's going to control the slides, perhaps.
Great. Hi, so I've trained in the more basic biological disciplines of cell biology and molecular biophysics. And my research focuses on understanding how cells sense and respond to information in their environment. So of course, your body is made of many types of cells that work together to build your tissues and your organs. And the conditions within your body are constantly changing. So there's lots of information that's constantly fluctuating in your body. So the cells of your body have to be able to sense this information about things like what nutrients are available or what hormones are currently circulating in your body. And in response to this information, they are able to adapt and change their behavior. So cells can constantly adapt to the fluctuating environmental conditions. So the next slide, please. Thank you. And so this process of sensing and responding to information occurs at the surface of the cell. On the surface of the cell, there's thousands of molecules embedded that act as molecular antennae that can sense specific information or signals in the environment. So the cell surface acts a lot like an old-fashioned operator switchboard, where there's lots of different signals coming into the cell, being received by the cell, and then being transmitted into the cell. And this process is highly regulated in normal, healthy cells. Cells only change their behavior when the correct signal is present. But I study how cancer cells are able to hijack this process and grow and divide and survive in an uncontrolled manner by ignoring a lot of the environmental signals. Yeah, so that's my very basic sort of broad research question that I'm interested in.
Great, thank you. So I think next up is Kavita Saran, who's one of our clinical investigators. She's at Stanford.
Hi, everyone. Yeah, I'm a dermatologist at Stanford University, and I'm an honored recipient of the DG Mitch Mitchell Clinical Investigator Award. And I studied the genetics and genomics of cancer risk and protection. So you all know that 1 in 3 Americans will develop cancer, 1 in 5 will die from cancer. But what you may not know and what I study is up to 10% of cancer occurs in people who are born with an inherited risk of developing cancer. And some of these you've heard of. You've heard of BRCA, next animation. You may have heard of Lynch syndrome, which causes an increased risk of colorectal cancer, or perhaps familial melanoma, where patients can have anywhere from a 60 to 80% lifetime risk of developing melanoma. But many of the genes that cause cancer risk are still unknown. Next slide. And I posit that the skin is the best way to uncover many of these genes that cause cancer risk. Well, why is that? Well, we have 35 billion cells in our skin that are constantly dividing, proliferating, and differentiating. And every time we go outside, you're getting exposed to ultraviolet induced mutagenesis. And so in many ways, you can think of us as walking mutagenesis experiments. It's not really surprising that we have 5 million cases of skin cancer a year just in the US. It's actually more surprising why we don't have more. And that's because we have a huge, huge network of DNA repair genes, tumor suppressor genes, checkpoint genes that are all there to protect us against skin cancer. So I study a cohort of individuals who develop extremely high numbers of skin cancer. And I use this cohort of individuals to discover new genes and characterize new genes involved in the skin cancer risk. Thank you.
Great, thank you. And then last but certainly not least, Jay Choi, who's another one of our clinical investigators. He's at Northwestern, who's going to talk to us.
Great. My name is Jay Choi. I'm a physician scientist. And so I have a truly translational research program where my lab is focused on trying to better understand and treat patients, the cancers of the patients I see in clinic. So I just wanted to have a first schematic to show that a lot of our focus right now is on T-cell cancers, which is a special type of lymphoma that occurs in the skin. It's called cutaneous T-cell lymphoma. So it's one of the types of skin cancers for which one of the cells are a T-cell, and that cell can become cancerous just like other cells in your body, like melanoma. And what we know is, unfortunately, as of today, most of the patients with this disease are all treated the same. But we know every patient is different. They have a different clinical manifestation, they have a different prognosis, and we certainly think that they have a different response to medications. And so I put a schematic here to try to make that easy for you, which is say that we think every person is different, and the molecular differences are represented by the different colors. And the different molecular aspects of people's cancers require specialized treatment, or this is a movement towards personalized medicine, which I'd like to bring to our patients. Next slide, please. So in our disease, we have basically found that the patient disease clinical characteristics are a function of 2 different qualities. One is the cell that it comes from, and the second are the molecular aberrations or mutations that occur in each of the cells. And so just like there are actually many, many different types of T cells, and so each of the different T cells can form different T cell cancers. And we have found that the source of the T cell that it comes from actually has a profound impact on clinical manifestations and prognosis. And this is combined with the mutations that they suffer to lead to specific molecular differences and clinical difference we find in patients. And I just show a quick example on the right, which is a survival curve, showing that people who come from the blue cell or the V delta 1 cell and the people with the red cell, V delta 2 cell, actually have completely different prognoses and really require different treatments. And this is the focus of my research.
Thank you very much. You know, maybe I'll begin by asking our awardees if they could try to give people a sense what is special about the Damon Runyon, and how is getting a Damon Runyon award impact you, even beyond, you know, the nuts and bolts of funding the work. But we like to think there are other components to it as well. So maybe I'll start with Kavita.
Yeah.
You know, what is it meant to be supported by the Damon Runyon?
Yeah, I think one thing unique about the Damon Runyon is, as Dr. Varmus mentioned, this mentorship that comes with this. I had actually heard of Damon Runyon. My mentor for my lab is a previous Jean Tang is a previous Damon Runyon recipient. And so I think, and she's just been an amazing mentor for me. So I think that that's one huge impact of the Damon Runyon, is the connections with colleagues, other Damon Runyon awardees, and alumni that have really helped to further our careers. The reputation is well known. It's extremely prestigious. Shortly after, a year after getting the Damon Runyon Award, I was promoted to associate professor. And I think that the Damon Runyon Award is one of many things, but definitely contributed to that as well. So I think there's just definitely this network of mentorship, and also it's well-regarded reputation that's been very helpful.
Great, thank you. Jay?
So I would echo Kavita's statements. You can look at this very concretely. I got the Damon Runyon Award, and shortly after, in the next 3 or 4 years, I was able to get another $4.5 million in funding from the NIH and other places. And every comment I got on my reviews is that Jay Choi is a Damon Runyon investigator. It's a solid investigator and someone who we should invest in. And I think this kind of mark of quality is something that really helps us very much so in the next steps. I would also just like to say that as the climate gets more competitive, most grants have become very conservative, and having a grant mechanism that funds bold science allows us to tackle big questions for which we're not sure what the answers are. And I think this is the way you make non-incremental advances. So this would not be possible without the Damon Runyon Award, for sure.
Thank you. And Lindsay.
Yes, yeah, I can sort of echo that and say, like, the project that Damon Runyon funded, the federal government said this project was unfeasible and unfundable. And so I would not have been able to do the work that I've done without the support of Damon Runyon. And this project did ultimately work and lead to really exciting discoveries. And I also want to add that one thing that I didn't appreciate before becoming a Damon Runyon Fellow was how beneficial being part of the Damon Runyon community was. They fund such a breadth of research, and we get to meet all the other Damon Runyon scientists and hear what they're working on the cutting edge of their fields. And so it's a really wonderful opportunity to learn a lot about what is really happening at the cutting edge across various disciplines related to cancer research. Great,
terrific. Harold, I'm frequently asked, what can we learn from COVID-19? You know, we've made very rapid progress there, and sometimes people ask me, well, are there lessons to be learned there? I do want to loop back also to our awardees in a minute, because I know it's affected their work at the bench. But, you know, from your perspective, what things can we perhaps learn from COVID, and what things are not as transportable?
Yeah, well, that's an interesting question, because, well, of course, all of us are dealing with this at different levels. And it's clear that at the completely pragmatic level, we're learning that certain habits that we have as scientists are habits that we ought to re-examine. Should we be traveling so much to meetings? Should we be doing more things by Zoom? And should we be giving more time to just sitting at home and thinking about what we're doing rather than frenetically doing experiments all the time? And I think that's a refreshing way to think about the practical impact. But at a deeper level, I think the public ought to have a clearer idea of how the science that came before we even knew about SARS-CoV-2 and COVID-19 made a difference in our response. You know, I'm old enough now to know that there was a time we didn't have any molecular tools that we were talking about. I lived through the AIDS pandemic, which of course is not over, but in the earliest stages, we didn't even know what the causative agent was for about 3 years, and it took time to have a test that was, and when COVID struck, within days the virus was identified, its sequence was fully published, people started making tests. Now there were tremendously difficult problems and errors in the way in which we implemented or executed the job that needed to be done to make testing part of a public health program. And I think there's a tremendous amount to be learned there and will be learned by a COVID commission and by people looking carefully at how we managed to make use of these remarkable tools that science has provided. And now everybody, of course, is exultant about vaccines because we've never seen a vaccine especially for a new disease, emerge so quickly. I can say a lot about that, but let's hear from some of the other panelists. I'd be curious to know what a younger generation that wasn't exposed to all this to times of what seemed to be, in retrospect, abysmal ignorance affects my own perception of how things worked. What did you all think about how we responded?
Jay, maybe I'll call on you. Would you be willing to give us a sense of what it's been like this year to try to get your work done, and then to Harold's question, how it may have colored your thinking moving forward about how we approach science?
Well, it's a great question. It's been very challenging for us because just as though the pandemic has closed down, or the shelter-at-home orders have shut down other businesses, that completely shut down our lab for many months. We've also faced some serious headwinds because some of the funding agencies actually ran out of money during the economic crises. And so I actually had less money coming in than I had projected. And then lastly, I had some people who were volunteers in my lab and they didn't have health insurance. I actually had to put them on payroll because I felt like it was really critical in the pandemic. So just to do the right thing by my trainees and try to keep things going, we've had to actually try to overcome these different things. But I would say overall, we've been resilient, and then things are going well, and we're going to continue our progress of trying to do the things right to make better cures for cancer. But it's definitely been difficult. The other thing that's been difficult is we've been trying to do shift work in the lab, and so that actually limits the number of hours anyone is in the lab. And so to compensate, we're actually trying to do things that potentially are more expensive, but actually take less time. And so all of these lead to a very complicated work environment. But that said, I think that a lot of us who do this work have a lot of faith in science. We believe that science can cure a lot of the problems that we're facing. And I think that what's happened recently is truly a testament to that philosophy. And so we're really excited about the rollout of the vaccines right now.
Kavita, I don't know if you want to chime in there.
I echo what Jay said as well. I would say one area that, when the pandemic hit, everything shut down, and one area that was hit particularly hard were clinical trials. So I had a Damon Runyon funded clinical trial that was in place when the pandemic hit and was shut down. And it did actually, we were testing a topical cream for the treatment of basal cell cancer. And we were able to actually pivot into a virtual only trial with the new use of technology and digital medicine and measurements. And it does, I think, will change the way I think about clinical trials going forward, having experienced now a virtual clinical trial, and whether or not, how often do we really need to bring patients in, and how can we reach a broader range of clinical trial subjects in this new digital age. So I do think that while it was very difficult for everybody, I think in science, there are some positives that will come out of it. And to answer Dr. Varmus' question, totally impressed with how quickly science was able to work. And it makes me feel very proud to be a scientist, just to even see the response in the vaccines.
And Lindsay, do you have anything to add?
Yeah, I think I'll just add, like, one takeaway for me from this whole year has been the importance for us as scientists to communicate what we do to the public and really help them understand how science works all the time, not just now. So that's something I'm sort of trying to do a better job at going forward. And I think also help train people working for me to do a good job at this as well.
I think the point Harold made is so important, however, that the reason we were able to go fast is because of the knowledge that had been generated, frankly, over decades because of investments in science. And that's really what Damon Runyon is all about, making sure we have that body of knowledge so we can move as quickly in cancer as we just saw with COVID. And the last thing I'll say, and then I think we're out of time, unfortunately, is my father-in-law used to say that A's hire A's and B's hire C's. And so I think part of the secret sauce of the Damon Runyon is we work really hard to find the A's. And I think we just heard from 3 examples of people who are really A's. And when we talk about funding brave and bold, we really mean it. So I want to thank you, Harold, for joining our panel. I want to thank our 3 awardees for our discussion. I'm sorry time is short, and I'm getting the cue that it's time to move on, but I enjoyed this, and I hope the rest of our audience did as well.
Thank you, Bill. Short but sweet.
Thank you.
Thank you all so much for a very inspiring and fascinating discussion. I'm sorry that we don't have more time to speak to all of you, but we do look forward to continuing the conversation with you and many of our other scientists during the virtual table, smaller discussions after this main program. So I am delighted to now introduce a very special guest, James Naughton. Jim is a 2-time Tony Award-winning actor and producer who has performed in numerous roles on screen and on the stage. His connection with Damon Runyon is twofold: through the Foundation's history with Broadway and through his personal connection to cancer, having lost his beloved wife, Pam, to pancreatic cancer in 2013. Jim, thank you so much for being with us today to share your story.
Hello, can you hear me?
Now we can hear you, yes. We can't see you at the moment yet, Jim.
You can't see me.
No.
Well, let me see if I can do something about that. How about that?
Yes, perfect. Thank you.
Well, that wasn't very graceful. I've made ungraceful entrances and exits before, but not virtually. Let me just say the reason that I'm here and participating is because I did have quite an extraordinary experience being the husband and caregiver of a woman who developed pancreatic cancer and fought it heroically, really, for 4 years. I was telling Jung some time ago that Pam had had a Whipple procedure at MSK, and then got all of her treatment, or oncology and chemo treatment, in Connecticut, which is where we live. And after about 2 years of chemo, my doctor, her oncologist, Dr. Richard Frank, in Norwalk Hospital, said, you know, we don't have anything else to throw at this thing. But there's a new trial that Pam might be able to qualify for that's going on starting soon down in San Antonio, Texas. Why don't you give them a call? So I called San Antonio, Texas, and I talked to Dr. Tolcher, and he said, I'd like to see her, and we'd like to see all the paperwork and all the tests. We sent everything down, and then we got on a plane and we went to San Antonio, Texas, and we got off the plane in August. And it was 114 degrees and blowing. It really felt like being in a blast furnace. But the good news was that she qualified and got into the trial, got the last place in the trial. And so every week on Tuesday, we flew, and there's only one flight per day. That's a nonstop between New York and San Antonio. On Wednesday, she got treated, and on Thursday morning, we were on the 7:30 flight back to New York. And we kept that up for 8 months, because when you're faced with something like that, you do whatever it takes. And we did whatever it took to try to keep going. After 4 years, and I must say that probably 3.5 years in, if you didn't know that she was sick, you wouldn't have recognized it. But in the last 3 or 4 or 5 months, it got to be very rough. And it made me say that after I lost her, that I would do whatever I could, whatever it took, to try to help find a cure for that disease. 3 years ago, her oncologist, Dr. Frank, who's also a researcher at the Ruggles Research Institute in Danbury, Connecticut, asked me if we could help him raise some money for a new trial to try to find a biobank, to try to find a biomarker, a blood marker for pancreatic cancer, which, as we all know, could be a game changer. So my kids and I, my son and daughter-in-law, all of whom are actors and singers, did a show. That's what we do in my business. We put on a show. We did a concert, and we kicked off that trial, and we got it started with $1 million that we raised that night. Last couple of months ago, I was up in Simsbury, Connecticut, to participate in the Ron Foley Foundation fundraiser. They're trying to fund research for cancer research. Tonight I'm here because I have a little history with Broadway, and I've always thought that the Damon Runyon Society research program was kind of unique because it gives 100% of the donations to the research. It doesn't go to administration. It also funds really young people, startups, trying to find the new scientific stars. And the other thing is that it focuses not just on one particular cancer, but on all cancers. So if you're thinking about making a donation tonight, I'd be really happy if you would. And now it gives me great pleasure to be able to introduce a video where you're going to hear from some of the young Damon Runyon scientific recipients who want to say thank you to you.
I am very grateful to everyone that donates to the Damon Runyon Cancer Research Foundation. It makes my research possible and helps fund the brave and bold discoveries that will change the way we treat cancer and help save thousands of lives.
Without scientific funding, advances in early diagnoses, chemotherapeutics, and treatments would simply just stop happening.
Having an organization like Damon Runyon believe in us and our science is incredibly uplifting, and it wouldn't be possible without your donations.
As a young investigator, I can tell you that funding at this critical juncture is really impactful.
Scientists like myself need funding in order to have the resources necessary to drive innovative research forward.
The whole point of research is that you don't know what the answer is going to be until you go and find it. And so funding continuously, broadly, for directions we don't yet know whether they'll pan out or not is so important.
New methods for prevention, diagnostics, and treatment for cancer will ultimately stem from rigorous discovery research.
It would be impossible for us to create this knowledge without the support and generosity of all of you out there.
Thank you very much. It's a great honor to be funded by such a prestigious fellowship. It's a great encouragement for the work that we're doing as well.
I study the fundamental question of how social setting affects disease progression. And without the funders and donors at Damon Runyon, we would have been unable to pursue these basic research questions.
So thank you so much for donating to the Damon Runyon Foundation. Your support enables me to pursue my passions. I'm incredibly grateful for everything that you're doing, and I hope that together we can advance cancer research.
Thanks to your generous support, we have raised nearly $1.1 million from this event. And 100% of every dollar raised will be used to fund the most brilliant young cancer researchers, because we believe that they will drive the innovation and breakthroughs needed to prevent and cure cancer. I want to extend a special thanks to our board of directors for their unwavering commitment to our mission. And thanks to the exceptional leadership of Alan and now Deb, I'm confident that Damon Runyon will continue to advance its mission of identifying and enabling the next generation of emerging talent that will make breakthroughs against all cancers. We thank all of our amazing Damon Runyon scientists, current and former. And if you're inspired by what you've heard today, we welcome you to donate at the link that's provided on your screen and in the chat box on Zoom. And this concludes our main program. If you have RSVP'd to join us for a virtual table with Damon Runyon scientists like the ones who just spoke, please refer to your email after this program concludes and click on the second link to join that special part of the event. In those rooms, our scientists will give you a brief summary of their current work and, time permitting, can answer questions. From all of us at Damon Runyon, thank you again for your support and your partnership. We are so grateful. Stay safe, be well, and we wish you and your families very happy holidays.